Hello everyone, it's nice to be with you today, even if through a more unique medium than usual. My name is Joyce Reimer. I'm a master's student of biomedical engineering at the University of Saskatchewan, and I'm pleased to share with you today the research of my colleagues and me on a cardiac cell simulation of a startle response in long QT syndrome 2. Here is an outline of what I will cover today. First, I will explain some background information on cardiac models and long QT syndrome 2. Then I will share the methods we employed to carry out the simulation, followed by the results and some conclusions. To start off, I will give you a brief background on computational cardiac models. There are a few different types of models that allow us to computationally simulate the heart. The primary ones include electrophysiological, biomechanical, and hemodynamic models. The one that I will focus on for this presentation, though, is an electrophysiological model. This is a more microscopic perspective of the ionic currents involved in cardiac action potentials at the cell level. Cardiac models in general have several uses. One is research on drug development or disease mechanisms. There are a handful of studies on the use of cardiac models in planning surgical procedures, and they also have a role to play in personalized medicine because of the idea that cardiac models may be customized to represent a specific individual's heart function. So cardiac models are gaining some traction for their potential use in clinical settings, but they are still mainly being used by the research community at this point. A few reasons for this are that there are many different types of cardiac models, and there are some discrepancies between them since they are all based off of different sets of experimental data. There will also be slight discrepancies in cardiac activity from person to person, since everyone has a unique mix of underlying factors. These make it difficult to represent a wide range of disease conditions with cardiac models. While this remains a complex issue, in our research we show that this is feasible to do and beneficial. In this particular paper, we showed this by simulating one specific disease, long QT syndrome 2. So long QT syndromes are a family of diseases called channelopathies, affecting ionic current flow across channels of the membrane. The name comes from the prolonged QT wave on an ECG, which is shown in the figure here, where the top ECG is from an unaffected individual and the bottom trace is from someone with long QT syndrome. The lengthened interval is caused by delayed repolarization of the ventricles. This can lead to a characteristic arrhythmia called torsade de pointe, as well as other symptoms including syncope and sudden cardiac arrest. Long QT syndrome may be congenital or acquired from certain pharmacological substances. Now, long QT syndrome 2, or LQT2, is one type of long QT syndrome that affects the rapidly activating potassium current IKR. It is caused by mutations in the KCNH2 gene that encodes part of the channel for IKR, and as you can see in the figure, most often the mutation affects the trafficking of the channel to the membrane, resulting in a lower density of functional IKR channels. Every long QT syndrome has a different trigger of arrhythmias, and LQT2's trigger is usually a sudden auditory stimulus, such as an alarm clock or some other sudden loud noise. If an arrhythmia is triggered, this may lead to ventricular tachycardia and possibly cardiac failure. A common drug treatment for preventing arrhythmia is beta blockers, but these are only effective for some individuals. So in this study, we conduct a series of simulations that are relevant to the challenges associated with LQT2. First, we simulate baseline LQT2 syndrome with a reduction in the IKR current. We then simulate a startle response on top of this to see if an arrhythmia occurs. Then finally, we simulate a treatment with calcium channel blockers, which is an alternative drug therapy to beta blockers that has shown promise for preventing arrhythmia in patients with LQT2. The mathematical model we used to go about this was developed by Grandi et al. In 2010. It is a system of 38 ordinary differential equations. These were solved for a single cardiomyocyte using MATLAB's ODE15S solver. We then adjusted parameters of the cell model until it matched experimental data. The reference study we used by Itzaki et al. Took measurements of induced pluripotent stem cells from a patient with LQT2. This data was then used to verify our model on the basis of IKR current density and the action potential duration up to 90% repolarized, or APD90. We also wanted to simulate a realistic startle response. In 2005, Reimer et al. Did a study on heart rate changes over the course of 70 seconds after a sudden loud noise. The plot on the right was taken from this study, and it was used as a way for us to determine the heart rate changes that might occur during the course of a patient being startled. These changes in heart rate were applied to the model by changing the pacing of the heart, which is normally administered at a constant frequency, to a piecewise function that changed frequencies over time. And these are the results of our simulations. First, this is a current density plot of the IKR in a healthy control cell compared with that of a cell with the LQT2-induced IKR block. As you can see, there is a reduction in the total current. This was carried out by changing the IKR conductance parameter in the model until a current density within the range of Itzhaki et al.'s measurements was obtained. This ended up being about a 65% reduction. To further verify the model, we looked at the action potential duration, because one characteristic of all long QT syndromes is a prolonged action potential. So at a baseline level, the action potential was only increased by about 8%, which you can see is the second curve close to the control. But when we adjusted the parameters to simulate startle, the duration extended quite a bit. So when an individual is startled, there is a sudden surge of adrenergic or sympathetic nervous activity in the body, which is partially causing the changing heart rate we saw in those plots from Reimer et al. A few slides back. To simulate this surge of sympathetic stimulation, we adjusted several aspects of the calcium dynamics. I won't go into these adjustments now, but if you're interested, they are presented in the paper in more detail. This caused the action potential duration to increase 145%, which is the dashed line on the far right of this plot. This number is within the range of those observed in Itzhaki et al.'s stem cells. This drastic change is a reflection of the fact that most individuals with LQT2 do not notice anything wrong until a triggering event occurs that causes an arrhythmia. In this case, part of the trigger was the sudden surge in sympathetic activity. On top of the sympathetic surge, we applied the changing heart rate and recorded traces of the cellular startle response over the course of 70 seconds. The top figure is the control and the bottom is with LQT2. As you can see, some action potentials appear to be skipped in the bottom trace due to the longer repolarization time. This slide shows the same traces just zoomed in. In the bottom trace, you can see that there are extraneous peaks between action potentials. These are called early after depolarizations and are considered to be a precipitating event at the cellular level for the characteristic long QT arrhythmia torsade de pointe, which can potentially lead to cardiac failure. Finally, these are the results of our simulation of calcium channel blockers, which are sometimes used to prevent LQT2 arrhythmias. So as you might guess from the name, calcium channel blockers reduce the calcium current in the heart, which is a current heavily involved in the sympathetic response. To simulate this, we adjusted the calcium parameters again to reduce ICA, and we found that this had a normalizing effect on the action potential duration. The plot to the left is from a few slides ago, but we added two curves that show the effect of calcium channel blockers on a cell with LQT2. As you can see, the action potential marked by the hollow circles is much shorter in length than the dashed curve on the right, which was the original action potential causing early after depolarizations. This result verifies that ICA plays a large role in triggering the LQT2 arrhythmia and provides justification for using calcium channel blockers in the prophylactic treatment of LQT2. And this is just to show that the full startle response with calcium channel blockers, which is on the bottom, is very similar to the startle response seen in the control traces at the top. In conclusion, we have shown that parameterizing cardiac models to represent diseases may shed light on the disease's mechanisms and characteristics. This study also demonstrates applications of cardiac models in clinical settings because we showed that we can observe a patient's disease under different conditions that may not be possible to recreate in person, especially something like startle or a sudden sympathetic surge. Also, because we showed that we can simulate particular drug therapies, this type of model may be used to determine the best treatment for a patient's specific LQT mutation. This has implications for the field of personalized or precision medicine. Finally, this type of model may be further used in biomedical research to study long QT arrhythmia triggers and their effects. That concludes my presentation. Thank you for listening, and please feel free to let me know if you have any questions.